Hi, everyone. I'm Madeleine Oudin, parent to Margot, who has SCN8A, who's 2 years old. And I'm also a researcher and who's recently started doing SCN8A research and getting into the community. And so I was invited today to share some of my perspective on exciting things that are happening in the SCN8A research space and what we have to get excited about and be hopeful about moving forward for new treatments for our children. So the first thing I think that was pretty cool this year was a review article that was published by leaders in the space in pediatric epilepsies called Precision Medicine for Genetic Epilepsy on the Horizon, Recent Advances, Present Challenges, and Suggestions for Continued Progress. And they talked about, 50 most commonly mutated genes that cause epilepsy in children. And then they focused in on SCN8A as really the poster child for the gene where it was the closest time from coming from that initial discovery of the gene back in 1995 by Miriam Meisler, then finding it in humans in Shea Hammer in 2012, to then 10 years later, having 2 drugs in clinical trials. And so, really SCN8A is seen in the community as the poster child, as the gene where we have this amazing advocacy, amazing researchers, and that we've been able to make so much progress. So that's really exciting. Another thing is, I think this year we've seen a lot of new people come into the SCN8A research space. So we have a really exciting study coming out of Sick Kids in Toronto, where Julia and Cam Kilner, parents to Maddie, who has SCN8A, raised over 120,000 Canadian dollars to run a study with patient blood samples to discover blood biomarkers for SCN8A. And this is exciting to kind of be doing that in a new direction. I've also started research in my own lab, and my husband and I have really recruited 3 or 4 labs to help us with this. So the community of people that are doing SCN8A is growing every year. And the more people we have working on this and trying to understand how this gene works is going to help us develop better treatments for our children. Then I think this year I had the opportunity to attend a couple of epilepsy meetings and really saw strong showing from the community. So many people working on SCN8A coming to the meetings, talking about their work, SCN8A mouse models being used. It was really exciting. I think it was really the DEE, the pediatric epilepsy, that was mentioned the most. And it was cool to see all the research that different labs are doing on how SCN8A regulates brain activity in different cell types in the brain, trying different treatments for it, building new models to study the disease. So there's really a lot of exciting research going on by some amazing scientists. And lastly, I think, we're seeing new genetic treatments that are coming on. We have some exciting work from Miriam Meisler's lab on antisense oligonucleotides. So these little pieces of DNA that can specifically alter the amount of a specific gene or change the way the gene functions. And so these are kind of a new way of treating diseases. And she had some really exciting data. Our daughter Margot is accepted in the N. Lorem Foundation as one of about 70 patients. They're also developing a treatment, an ASO treatment. And so, any data that we can get from treating an SCN8A patient with an ASO, will be really valuable for treating more kids. So that's exciting to see this technology being applied to SCN8A and seeing, how this will then impact the patients. And I think looking around too, this year, there's a new clinical trial that started for Dravet, which is SCN1A. So it's a different gene, but it's still sodium channel. And this is using an ASO to try and correct the disease. So this isn't no longer just treating it with drugs, trying to, stop the symptoms. This is actually the closest thing to fixing a mutation in a sodium channel that can cause epilepsy. And this trial has shown some really interesting results. It's still ongoing, but I think there'll be a lot to learn from that and how these genetic treatments that, again, we're trying to fix it, not actually just treat it, whether, how much this can help kids with their seizures, but also with their developmental delays. So I'm really excited to see this trial go on and that we can use what we learn to help our kids with SCN8A. So those are some of the reasons that I think seeing everything that's happening in the last year in this space make me really hopeful as a scientist and as a parent. And I'm excited to see where we're going to go next. Thanks.